Once you've set up and run an experiment on Rosalind, you end up with a series of screens that allow the ability to explore your results. We're going to quickly touch on that here. This is the experiment home. This is the summary of the experiment that was set up here. And this can be accessed by clicking on any experiment, either from search or within the navigation or from a project space or from a library. You always end up in this place when opening up an experiment. A couple things you'll see right as you come in. We can immediately note that we are viewing an experiment. The title of the experiment is displayed here underneath, as well as right here, the full name. A nice description of the experiment that was added at the time of the experiment setup. This can also be edited after the fact, and some key details. This was sequencing. It came in by RNA-seq process counts as opposed to FASTQs, which are also possible. It's human and was built run on genome build GRCH 38. There are 32 samples. There are 3 primary attributes: time course, cell line, and treatment. And I'm the experiment owner for this. Below, we have the full method section that's ready to go. You can just copy and paste that out and drop it in any publications, all set. I can see this is shared in 2 collaboration spaces, and there are a couple related experiments to this. And this is the experiment summary, which is present on this tab. By clicking on the sample tab, I get a listing of all of the samples and the metadata that is associated, or the sample annotation, for every single one of the samples in my experiment. Now, notice I can add attributes and enhance the sample annotation as the owner of this experiment at any time. So if I set up my experiment and I forgot to add time course or cell line or some very important aspect that really differentiates the biology or some way for setting up comparisons, I can just click add an attribute, which will require me to download the attribute file, add that in Excel, just add a column, add the relative values for every sample, and then re-upload that new file. And that will update the metadata within Rosalind for any existing experiment and allow new comparisons to immediately be built. Also, I can see the file download here. There's a nicely organized collection of data, my source files, the individual attribute file, the counts matrices, both raw counts with annotation, as well as normalized counts, and a summary of all the genes and the associated annotation for every comparison that's created. So if you want to work with the comparison data and you want to do that offline, you can easily just click download and grab that comparison data. Or one of the great capabilities, especially for folks with enterprise subscriptions, is the ability to have unlimited process counts experiments. So here I can download my raw counts and my attributes file. I may choose to modify some aspect, either remove a sample or do other changes. I can merge those files together and create a brand new experiment. And these files are already set up and ready to go in a beautiful format that makes it easy to manipulate the data. We've already been working with our comparisons. You can see a full listing of all comparisons here under the comparison tab. And this is also where I can go ahead and add comparisons or add meta-analyses right here. There's a few QC plots that enable a big picture view. This is a counts-based experiment, so there's a limited set of QC data that's available. First is my correlation heat map that allows me to look at the groupings across my samples. So we can see my 3 different cell lines grouped beautifully in this experiment. And the violin plots showing the distribution of reads across all samples. Again, very, very clean view of the information. Everything's downloadable. And lastly, the MBS plots, which allow me to look at the grouping of my samples. Here we see by cell line. So nicely grouping across each one of those. And if I want to download, I can just click download and I'll get each one of these images, and or I can scroll and see each one of these. So a really great way to quickly look at the QC.